All right, we still have more people coming in, but we're going to go ahead and get started. My name is Michael Myers. I'm the Chief Communications Officer for the Diabetes Research Institute Foundation. And I want to thank everybody for their attendance tonight. It's a very exciting night. We have two wonderful scientists and individuals joining us for this evening's conversation. This is another in our series of Meet Our DRI Scientists. And it's a great opportunity for everybody to have a look under the hood at the exciting work taking place at the DRI on a daily basis, work that could not take place without the support of many of you out there and many others across the nation and around the world. And without that support, the great work that you're going to hear more about this evening couldn't take place. So we thank you. Without further ado, I'm going to introduce our guests and just a couple of housekeeping items before I do that. Please mute any phones or cell phones, etc. Your lines will be muted. If you have any questions you would like to ask, please submit them via our chat, and we will try and get to as many as possible. We've already had a number of questions submitted in advance that we will be addressing up front. If your questions do not get addressed during the live call, I assure you we will be providing responses with our two scientists to those questions by the end of this week. So please do submit any questions you have via our chat. Without further ado, Norma Kenyon is the epitome of passion, empathy, resilience. Norma Kenyon is the epitome of passion, empathy, resilience, and a tireless dedication to our scientific mission to find a biological cure for type 1 diabetes. Dr. Kenyon is a deputy director and the Martin Kleinman Professor of Surgery, Microbiology and Immunology, and Biomedical Engineering at the Diabetes Research Institute at the Miller School of Medicine at the University of Miami. She earned her undergraduate degree from Duke University and her PhD from Virginia Commonwealth, followed by postdoctoral positions at UCLA and the University of Miami. She left academics to work for Coulter Corporation early in her career, but ultimately returned following the type 1 diagnosis of her young daughter to focus her career on the pursuit of cure-focused type 1 diabetes research. Dr. Kenyon has served as chair for immunology in the NIH-funded Type 1 Diabetes TrialNet and the Clinical Islet Transplant Consortiums. She was a member of the National Advisory Allergy and Infectious Disease Council and recently finished service as a member of the NIH Council of Councils. In addition to her academic pursuits, Dr. Kenyon has served as a scientific advisor to the Food and Drug Administration and a senior associate dean for translational science at the Miller School of Medicine. Today, Dr. Kenyon and her team lead pioneering work in immune protection for cell therapies, advancing approaches such as anti-CD40 ligand-based therapies and cellular strategies to prevent rejection and enable islet transplantation without lifelong immunosuppression. The name Camilla Ricordi is synonymous with diabetes research worldwide. Professor and Chief of the Division of Cellular Transplantation, Department of Surgery, Director of the Cell Transplant Center, and Director Emeritus of the DRI. Dr. Ricordi was born in New York and received his academic training in Milan, Italy. After military service in the Italian Air Force as a medical officer, he worked at Washington University in St. Louis and at the University of Pittsburgh before moving to the University of Miami in 1993, where he served as professor of surgery, medicine, biomedical engineering, microbiology, immunology, and director of the Diabetes Research Institute for 3 decades. Since 1993, Dr. Ricordi has served as medical director of the NIH-funded DRI CGMP, Current Good Manufacturing Practices, Advanced Human Cell and Biologic Product Manufacturing Facility, which has provided an invaluable service to centers throughout the United States and worldwide by offering training programs and distributing human tissues, cells, and other biologic products for research and clinical applications. Acknowledged by his peers as one of the world's leading scientists in diabetes cure-focused research, cell transplantation, and regenerative medicine, Dr. Ricordi is well known for inventing the Ricordi Chamber. This year, we celebrate the 40 years of the Ricordi Chamber, and an automated method for islet isolation that made it possible to isolate large numbers of islet cells, insulin-producing cells, from the human pancreas. The method was eventually validated at Washington University in St. Louis, where he served as the NIH trainee. Recruited to the University of Pittsburgh Transplant Institute in 1989, Dr. Ricordi led the team that performed the first series of successful clinical islet transplants of donor pancreatic islets, reversing diabetes after their intrahepatic infusion. The procedure is now used worldwide by centers performing clinical islet transplantation. Dr. Ricordi is driving the next generation of beta cell replacement therapies, integrating stem cell derived islets with innovative transplant platforms and immune tolerant strategies to move us ever closer to a cure. Doctors, the floor is yours.
Great. Thank you, Michael. That was our talk and time allotted. Now we're open for questions. It's a great thank you all for being with us. And Norma, Kenyon, and I are really pleased to give you this little update on what is happening worldwide and where we stand in the race on the path to a cure. And I'd like to start briefly touching this because you already
You suggest an islet cell on a transplant? You don't have cells. You need a Ricordi chamber.
Dr. Camillo Ricordi has been leader in the islet transplantation from the beginning. He developed the islet isolation technique, which allows us to even perform the islet transplantation. He's been mentor not only to me, but to many scientists from the States and other countries.
Many congratulations on 40 years of the Ricordi Chamber. You share all of your protocols for islet isolation. It wouldn't have been possible to achieve the clinical results we got with the Everton protocol if it hadn't been for your Ricordi Chamber. Thank you for giving the Ricordi Chamber to the world.
Thanks to your chamber, we've done 1,394 isolations and 310 transplants.
Professor Ricordi, for his 4 years of dedication bias, you have a sum up. Thank you.
I'd like to thank Professor Ricordi on behalf of people with type 1 diabetes, because your invention has changed their life and also saved the life of people with type 1 diabetes.
through making this available worldwide, we had everyone collaborating to improve the method.
We just said, this is something we have to do, and we do it. We all have the dream of finding
2005.
I stop here because there are all patient witness that people living without insulin for over 20 years, and it's too long. But I wanted to stop on Ramon Poe because he has been really the master and commander behind the distribution of this chamber worldwide, and created through his family support the facility in BioRep Technology, who was able to manufacture all the equipment we are using in Miami, and we made available at cost worldwide. So I renounced to all the patents, like I was inspired by Banting and Best that released the patent of insulin for $1 nominal. And then I said, I'm not pretending it's as important as the patent on insulin. I said, islets don't belong to me, belongs to people with diabetes, and we distribute equipment, protocols, training, and everything worldwide. So I want to thank Ramon Poe on behalf of all of us at the DRI and worldwide, because he's been really instrumental to make this technology available in all continents. And here in one slide, for years, like we started with the ability to extract islets from the human pancreas, then we reverse diabetes in preclinical models, both fresh culture cryopreserved islets, and we did the first successful transplant in 1990 at the University of Pittsburgh. And then you fast forward all the way to the phase 3 trial of FDA phase 3 trial supported by NIH at 8 institutions in North America. But at the same time, with Norma Kenyon, we'll talk about the anti-CD40 ligand, what is now the Eledon trial that we are conducting, thanks to Piotr Witkowski at the University of Chicago, in a major collaboration with the University of Chicago and now University of Miami. We are happy that we were able to still focus on elimination of the most toxic anti-rejection drugs. And then you'll hear about other strategies that we are addressing going forward. So the first message is that over the years, we have highlighted how the quality of the islet product our islet product at the beginning of the islet transplant procedure is really key. So if you don't have good pancreas and good islets, the results long-term are not very good. But if you have good islets, and often we have to get pancreas that are discarded from every organ transplant, but when you have a good quality initial islets, the results at 20 years, you see here in blue, are even superior to those of pancreas transplantation as an organ. These are fantastic results at the Edmonton group with James Shapiro that were published recently. We also showed from our experience at the DRI, but also in many other centers worldwide, that the survival of patients undergoing islet transplantation, despite the need for anti-rejection drugs, is quite remarkable, like around 85% at 20 years. And this is an age range where diabetes start having a hit. So these results are actually superior of those that you can expect with insulin without immunosuppression. So in many cases, islet transplantation can today be considered a life-saving procedure. Now the big next quantum leaps, and where we're all focusing now for many years, is that we need to eliminate toxic immunosuppression and then develop adequate sources of insulin-producing cells. But that will be necessary when you will eliminate immunosuppression, because if you need chronic anti-rejection drugs, we have plenty of islets available from donor, because we have 14,000 donors a year available, so that after successful transplantation without immunosuppression, then we'll need to prevent disease recurrence after an initial successful cure. So that's why we focus so much today also on making sure that after someone gets an islet transplant, remain focused on health and health span, and don't just think you can just go out and eat pizza and ice cream like there is no tomorrow, but we'll keep attention of all the principles that everybody should follow to avoid falling in the trap of chronic degenerative diseases that unfortunately afflict over 90% of people in the United States after age 60. So Norma, this is your time now to show what amazing work.
Thank you, Dr. Ricordi. It's great to be here tonight. I have to admit, I got a little emotional. I saw a couple of friends from high school in the participants, and Cheryl, I know you're there. So it's great to be here with all of you tonight. This is a slide that basically encompasses 30 years of work that Dr. Ricordi and I started many years ago at the DRI. We had the opportunity, our focus has been that for this entire time, how do we use safer, more effective anti-rejection drugs so that you don't adversely affect other organs, you don't suppress the function of the islets themselves. And so we've been working on many different approaches, and we heard about this antibody back in the mid to late 90s that targeted a molecule on T lymphocytes that caused the rejection. It's an early molecule expressed by cells that recognize the transplant, and it sounded like it could actually be one of the agents we've been looking for to stop the cells that are turned on by the transplant, but maintain your other cells effective to prevent inflammation. So we were fortunate to try it. It was a collaboration with Dave Harlan and Alan Kirk at the Naval Medical Research Institute at the time, and Biogen, who had this drug 5C8, developed by some basic scientists back in the day. The results were extraordinary and first of its kind. Dr. Ricordi, if you can just show that top graph there. When we first tested it for the first time ever, we were able to show, so on the bottom axis there, that's the days after the islet transplant. On the left axis, that's the blood sugar level. And what we were able to see is that in a clinically relevant preclinical model, we got a safe and effective long-term survival. Prior to this work, the longest islets had survived in a model such as this was 100 days. And we were able, actually at 800 days, we stopped giving any drug at all, and we had another 500 days without any immune intervention. So this drug had tremendous potential. It was actually being studied all over the world in different autoimmune and other indications. But in some patients in those clinical trials, there were thromboembolic or blood clots occurred, and the drug was stopped. It was kind of devastating. I personally thought that my daughter would be cured very quickly. And so Dr. Ricordi and I never gave up. We continued to try other approaches. But over time, we made a lot of progress, never quite hit where we were with anti-CD40 ligand that so safely and effectively prevented rejection. Dr. Ricordi came to my office one day, told me he'd heard about this new antibody of Analyxis Therapeutics that's now Eledon Pharma. I said, that's great, go call them right now. So we were very fortunate to take the new AT-1501, now called tegoprubart by Eledon, that's being used clinically, and we were able to show that the drug was very similar to what we had tried back in the late 90s that had the tremendous results. And the level of excitement is Islet, but also it works in many other indications. So we were able to quickly work with Steve Perrin at Analyxis and now Eledon and demonstrate that we had similar results to what was obtained with the original 5C8. All of our preclinical data, including the older data, now this new data was used to enable FDA approval of the ongoing clinical trial, which we'll give you a snapshot of that next. So here you can see the funders for this trial and the participants being led by University of Chicago with Dr. Wachowski in collaboration with Dr. Ricordi and I in Miami. Next slide, please, Camila. The protocol, the treatment protocol, I won't spend a long time on this. I know it looks way too scientific, but essentially in islet transplantation, we give a drug early on to target the T cells, to calm them down before we do the transplant. But the thing to recognize are the blue arrows on the top line. That's the timing of the dosing of the tegoprubart, which follows what we developed in our preclinical model at the DRI. And we have a little bit of data that we can show you next. And this is just one of the patients. There are a couple more, but I'll focus on this one. That in the past, we've needed a lot more islets to get patients off insulin. What we did is the same thing we studied in the preclinical model, which I neglected to say in my excitement, is that we were able to eliminate a drug called FK506, which is toxic to islets and kidneys, replace it with the anti-CD40 ligand in our preclinical model. That's exactly what we did here. It's working very well, safely and effectively enabling patients to come off of insulin. And I think you can see that on the top blue slide there. You see where the patient starts the hemoglobin A1C, the numbers and how it comes down. Below the dotted line, the patient is insulin free. And those results on the right are metabolic challenges showing that the function is still doing extremely well a year and a half out on this patient. So Camilo, I don't think we need to go into too much depth on the next two, but in other patients, we're seeing very similar results. So if you want to go to the summary slide, Camilo. 10 out of 10 patients beyond 1 month post-transplant are insulin-free. Dr. Wachowski, I should say, just presented all of this data in Barcelona last Friday at a big diabetes technology meeting. There have been no unexpected adverse events or severe hyperglycemia, which is something we try to avoid, no opportunistic infections, but really importantly, no signs of kidney and neurotoxicity that historically observed on FK506. So our goal to more safely and effectively prevent rejection and enable engraftment and insulin independence is appearing to be met. Next slide. One of the key things that we showed in our early studies and that has borne fruit is that when you use an agent such as anti-CD40 ligand, tegoprubart, that isn't toxic to the islets, you get 2 to 3-fold higher function of the insulin-producing cells than you do with traditional immune suppression. So better function, safer, more effective. And with this, I'm going to turn it back over to Dr. Ricordi to tell you about our next clinical adventure with tegoprubart.
So thank you, Norma. This has been very exciting, like 10 out of 10, and with much higher islet function because you eliminate Prograf tacrolimus. You have to understand, is a drug that is so diabetogenic. It can cause diabetes in a patient that doesn't have diabetes to begin with. So in kidney transplantation, person without diabetes undergo this immunosuppression and then require insulin, and it's called transplant diabetes. So you know how you have to use a drug that prevent immune attack of the transplanted islets or organs in other cases, but then it increased the metabolic demand on the pancreas or on the transplanted islets 2.5-fold. So then you go into exhaustion, and that's the reason why transplant on Prograf immunosuppression have been generally fading. You see that at 10 years you have 50% still working, and less than 20 years, but still has been a remarkable results overall, improving the life of patients on program immunosuppression. But now the big message that with this Eledon with anti-CD40 ligand and these results, you can now include in clinical trial patients with initial kidney dysfunction. So you have to understand it in all clinical islet transplant trial before, if you have kidney dysfunction, if your glomerular filtration rates start decreasing, you are excluded, was an exclusion criteria. And also if you need insulin above 45 units a day or more than 1 units per kilogram, you would be excluded because we knew that the islets cannot support that kind of insulin requirement. Well, now all this restriction will be lifted with this new immunosuppression protocol that is more immunomodulation and less toxic immunosuppression. So that is the big step forward in this trial, that also patients with chronic kidney disease stage 1 to 3A will be included as potential candidates. This is on the other fronts, besides anti-CD40 ligand, there have been a lot of activities in stem cell-derived islets. These are studies we conducted, been working with Doug Melton and Felicia and the whole team since SEMA Therapeutics and now became Vertex. There is an international trial ongoing between North America and Europe, and the initial results were very promising. There was a parallel study called VX264 with a macro device immunoisolated to try to protect the islets without any immunosuppression that unfortunately has failed. So far has been still a challenge to try to protect islets with micro or macro encapsulation technologies. But the trial is moving forward to phase 3 with the standard immunosuppression. So I hope that Vertex will even consider saying, guys, take Eledon or acquire or collaborate, because if they move from Prograf to anti-CD40 ligand, I think even the results in stem cell-derived islets will be much better. Then we've been collaborating for decades also with China. That was one of the first centers worldwide to join the Diabetes Research Institute Federation. We trained them for islet transplant, and they performed the first successful islet transplants in Asia decades ago, and now they're sort of moving forward much faster because there is a regulatory system that we may discuss later in the discussion. But China has a path that allows them to move much faster, testing new potential breakthrough strategy on the path to a cure for diabetes. So this one is one of their initial publications where they show fantastic results. Patient of insulin is now over 1 year and a half, almost 100% time in range, and more importantly, hemoglobin A1C down to 4.76. That is quite remarkable in islet transplantation. But also what is important to see is the time in tight range, that is levels between 70 and 140, also close to 100%. So this is quite spectacular. If you see people reporting only time in range, the level of glucose could be 70 to 180. So if you wake up in the morning with 160 and you're not going above 180, you're still pretty good as far as time in range. But that is why it's so important now to show both time in range and time in tight range. And this is a slide that I asked them to provide us to point out the fantastic metabolic control they can achieve. At the same time, the rival group in China that we also train in Miami from Shanghai, they trained with us in 2018, and they already did 310 transplants in Shanghai, and they published very recently also a paper showing both in type 2 diabetes and type 1 diabetes a complete insulin independence and a fantastic technology. Both groups use peripheral blood mononuclear cells, like just a blood sample from your blood, and then transform it into inducible pluripotent stem cells that are differentiated towards islets. And this group in Shanghai has the ability to produce 1,000 therapeutic cell doses from one master cell bank at a very competitive price, because what we're addressing now is not just the safety and efficacy of a treatment, but is also the affordability. Can we scale it up, and can we make it affordable so that everyone can actually benefit from an eventual therapy. And that's why we've been very pleased on seeing the progress of these collaborators worldwide. This is the last paper that I just published in February 26, just weeks ago, on the autologous and allogeneic stem cell-derived islet therapy with complete loss of endogenous beta cell function pre-transplant, and we are very happy to be working with them. These islets are also called e-islets because they come from the endoderm. They prevent the risk of teratoma. That is also always a concern when you use stem cell-derived islets. Are like really remarkable because in a way is a way to go up in the differentiation and eliminate the probability that cells can become some other lineage like mesoderm or become cartilage or some other tissue. So this has been preliminary results, but very encouraging. So you'll see a lot of hype online, like China cured diabetes, type 1, type 2, like actually it's still requiring immunosuppression, is a great step forward, still need to be validated, and hopefully will be confirmed, validated, and we can continue to collaborate going forward. Another exciting results has been presented and published by the Uppsala team with Sonia Schreipfer, that has been the master and commander of the gene editing technology to make islets stealth to the immune system. And there was one of the co-founder of Sana Biotechnology that presented this initial clinical trial. Is one patient, one example, but has been remarkable because it proved the proof of concept, even if the C-peptide was very small amount, as you will see in a few slides. It showed that you can protect islets from immune destruction. The caveat is that in these patients, this initial technology by Sana could not protect from autoimmunity against zinc NT8. That is one of the autoantibodies that can be present in 60-80% of type 1. So it's like protection from autoimmunity, yes, but from autoimmunity incomplete. And then I'm very happy to report that we are now working with Sonia Schreipfer, that team up with myself and James Shapiro, and left Sana Biotechnology because she developed a new breakthrough platform for gene editing that will hopefully allow us to not only protect from autoimmunity, but also from autoimmunity from any kind of autoantigen. So stay tuned, hopefully in the near future, for results in this direction. At the same time, we had work on bioengineering and intra-abdominal endocrine pancreas with the first successful. This was the first ever successful islet transplant in a site that is not the liver, where islets have been traditionally transplanted. And is important because the liver is not retrievable. So if you put stem cell-derived product, you're more concerned. You have to make completely sure that they're safe besides being effective. But the omentum is this curtain that we have in the abdominal cavity that protects all the organs that react in case you have an accident or a stab or a wound. And you see it here in this laparoscopic minimal invasive procedure. You can actually seed the islets, fold the omentum, and you create a pocket that drains like a pancreas to the liver. So it's complete physiologic, and we were able to have the first successful cases. But the reason we developed this system, and this you can see the blood glucose after transplantation in the omentum that are quite remarkable. But the reason we did it is because we wanted to then develop strategy to eliminate completely immunosuppression by localized immunosuppression and anti-rejection strategies. So that's why we have been helping in the past years iTolerance, that has developed a strategy that was developed by Ander Garcia and at Georgia Tech, and Haval Shirvan and Esma Yolko at now at the University of Missouri, that is a combination of a streptavidin Fas ligand that is practically is a molecule. The Fas ligand is a molecule that tag any immune cells that get activated, express Fas, that is this molecule, and become susceptible to linking the Fas ligand that will induce death of these activated immune cells And by doing that, will increase the number of regulatory T cells. So if you see, this microgel have been engineered so that they look a little like a COVID virus with the spike protein on the surface, but in reality it's much bigger. It's bigger like an islet, like 200 micron diameter, and the fast ligand is the red dot. So this is like sort of a mine that you put at the transplant site to intercept any cells that is activated and kill it in a way that doesn't induce inflammation, is so-called apoptosis, and promote regulatory cells at the site of transplantation. So you combine these microgels, immunoregulatory, with the islets here at the transplant site. The red cells are the bad cells that come to kill the islets. They get intercepted and they get killed. These are kind of iron dome around the islets. And then by doing this, you leave and proliferate the blue cells that are regulatory T cells at the site of transplant. And importantly, this is just at the site of transplant, so you don't alter immunity systemically. So you protect the cells in the transplant, but you maintain the defense against any other virus, bacterial, tumor possibility in the body. And this has been advancing now to preclinical studies to enable the first clinical trials, hopefully in the next year. But the results have been quite spectacular, with showing that you can stop completely all kind of immunosuppression, not just Prograf, after a transient therapy with rapamycin. So we are very excited of this potential therapy as well. Now, if you put all the peak stimulated C-peptide obtained with different strategy, you see that the Sana first patient was just a proof of concept, was very little C-peptide, was achieving like sort of 20 picomole liter stimulated to get completely insulin dependence. The Vertex or the Endocell trial had over 1,000 picomole liter, around 1,200, and the Beijing Tianjin group, that is the other slide that I show you with 100% time in range and time in tight range, they have achieved 2,500. And in the Eledon trial, now you remove Prograf, you remove toxic immunosuppression, and boom, you are like up to level that we never saw before. And this is important because, as you know, some islet transplant require more than one donor. Like in the Edmonton trial, you had to use 1, 2, 3, and even 4 pancreas donor. But now this is a strategy that I'm convinced moving forward will allow us to do one-to-one islet transplant in a very successful way. Now, I just want to conclude raising an element of concern and also an opportunity for intervention, because we can do 100 transplant a year, 1,000 transplant, 10,000 transplant, 100,000 transplants successfully every year. But we are dealing with a tsunami of pandemic of chronic diseases that affect especially American after age 60. But if you look at the diabetes numbers, there were 171 million of people affected in 2000. Today we are 590 million, projected to become 853 millions in the future. So clearly we cannot win the battle just concentrating on treatment with those with the most severe form already affected or reversing disease progression. That's why we are having international collaboration now, and I'll be speaking in Washington March 26 with the leadership of ARPA-H, Milken Institute, and Italian and American healthcare leadership to see how can we also work together to predict risk factors that can lead to autoimmune diseases or other diseases, and intercept and neutralizes with disease prevention, defeating them before they can progress towards the disease. Of course, we will not abandon those who start being diagnosed or have the most severe cases, but also concentrate with the same strategies as in 1 in 4 to prevent disease recurrence after an initially successful cure. This is very important because I always tell our friend and patient recipient of islet transplant, if you are off insulin, don't start celebrating and having ice cream and pizza like there is no tomorrow, because that no one should do it, whether you have diabetes or if you don't have diabetes. So that these pandemics that we're seeing today between autoimmune diseases that affect 20% of the population with 100 different pathologies, severe cases of COVID or viral diseases and chronic diseases associated with accelerated aging have a lot of common risk factors that we can identify, intercept, and defeat. And I even wrote a couple of books to address these issues. And I would like to thank all our multidisciplinary fantastic team at the University of Miami Diabetes Research Institute and the CGMP, now NAP2. We have the NAP2 Regenerative Medicine Center and CGMP facility at the DRI, thanks to the support of this amazing organization that has helped the Diabetes Research Institute Foundation over the decades. We have a fantastic team, regulatory CGMP, clinical immunology, immunogenetic model system, biomedical engineering, stem cell regeneration, reprogramming, and also this DRI Federation and international collaborators that there are too many to list. We have over 20 centers worldwide, but we are working, helping with the same enthusiasm as intramural programs and our own investigator, as well as people worldwide, and as far as they commit to help us reach a cure of diabetes in the fastest, most efficient way possible. This has always been my philosophy over the decades and will continue to be so at the Diabetes Research Institute. So I thank you very much for your attention. Thank you, Norma, for your amazing contribution, and everyone else at the DRI and the foundation, and we'll be happy to try to answer some question if they're easy enough.
Thank you. Thank you both. This was tremendous and very, very exciting news for a lot of people. And I can tell from a lot of the comments that have come in. We've had a number of questions submitted, but before I get to the questions, a young woman named Katie Beth Hand, who's going to be visiting us this Friday at the Institute, just sent me a text. She just sent me a text a few minutes ago that I would like to read briefly, if I may. Doctors Kenyon and Ricordi, thank you both for the many years of hard work, dedication, and research that went into making islet cell transplants like the one I had with Dr. Witkowski in Chicago, January 13th of this year, possible. The work you have done has been truly life-changing. Because of your efforts, I am living a happier, healthier life. I will be forever grateful to you both. I think we clearly have our why there in Katie Beth's beautiful words. So I just wanted to share that with everybody. We look forward to meeting her on Friday.
Happy that she's coming tomorrow, actually, and then I'll take her to the Institute Friday, and will be a fantastic opportunity to share her experience. And she's been also an amazing supporter and advocate for the campaign that we are now doing for reclassifying islet transplant as organ transplant, because right now we've been completely blocked by outdated regulatory environment from the last century kind that, unlike China, France, Canada, Australia, is blocking us. And you will hear, probably you already know much from social media. Devin Holt has been posting amazing interview, and we have now letters coming from Breakthrough T1D, from the Diabetes Research Institute and Foundation, from NAP2, from the North American Building Trades Unions. Just a letter arrived to Secretary of Health RFK Jr. As of today, so that we are all united front to try to make America competitive again, if you want to use a popular slogan nowadays. But is really so important that we train the rest of the world. We're working as a team, collaborative, and then you see that before the finish line, we're sort of stop, and then you see China, France, Canada, Australia. So it's so important that we are allowed to be competitive and contribute to the next quantum leaps, definition of new strategy to eliminate completely immunosuppression, develop unlimited sources, and the whole difference is that we have only this one track here, and islets are regulated like drugs, and so you need to be treated like a manufacturing drug company if you want to do islet transplant. The cost becomes incredible, and it's just blocking everything. I feel so sorry that we train the rest of the world. France is now approve a standard of care and reimburse UK network, started a network of centers in the UK based on our results in the United States, funded by American taxpayer and NIH. And China did 310 cases in the same time we did 10. And it's a pity we cannot allow to drop the ball. As I always say, like we did the first base with the automated method, second base with the first successful trial, third base now with replacing the most toxic anti-rejection drug. But we cannot stop before the home run, which is a cure. And if someone is trying to drag us down, letting everybody else passing us to the finish line, I don't care, because I don't care where the cure will come from. We're going to celebrate no matter what. But I feel sorry for academic centers in the United States and our patients if we are cut out of this progress in such a way. So I really hope, and I'm confident that now with the support of Aaron Kowalski and Breakthrough T1D, the DRIF, NAPTO, everyone is on the same line to try to make this difference happen and allow islet transplantation to move much faster on the path to a cure.
Thank you, Dr. Ricordi, for those eloquent words and a very important issue at hand. So we have a number of questions. We want to make sure we get to as many as possible. And for those we don't get to on this call, as I said earlier, we will respond to your questions within the next couple of days. So feel free to include them in the chat. First, a couple of questions, one for each of you individually. Dr. Kenyon, your work over the years in anti-CD40 ligand overcame many hurdles, as you mentioned some of them, to help create the very promising islet transplant results we are seeing in current trials. How do you motivate yourself to continue the fight against seemingly insurmountable odds?
Wow. Okay, that's a tough question. I'm a driven person in general, but I will say that whenever times get tough in certain areas, I do have a personal connection that is very close to me and I look back to, and that's my daughter. But it's not just my daughter. One thing I did want to say is that the real heroes are the patients. And I know Dr. Ricordi agrees with me. We have some of our previous transplant patients, and I can see our participants, and we have Katie Bath on, and we're just so grateful for them for taking the chance on us and the work we've done. So I'm inspired by people with type 1 diabetes and their families and their stories. And I also know that the work we're doing has the potential to help not only those we are close to, but many other transplants and conditions worldwide. So the science is exciting. When it gets tough, I look to the people I've met.
Obviously. Dr. Ricordi, if you can point to one thing, what are you most proud of with regard to the Ricordi Chamber and its impact on patients and families, as well as cure-focused diabetes research?
Well, actually, when I was inducted in the National Academy of Inventors, with Bob Langer and other much more important inventors than myself, I was asked a similar question: what is your most important invention of your 117 invention disclosure that paralyzed Norma's team at tech transfer at the University of Miami over the decades? But I said the most proud element for me has been sharing worldwide and giving up the rights of the royalty and the commercialization of the technology that I developed to make it available worldwide to all centers that want to help us reaching a cure in the fastest, most efficient way possible. And I've been said, well, you're utopistic, you're crazy. A friend of mine, president of a pharmaceutical company, told me, you think you're smart, and now if you need 100 million for doing a phase 3 trial or a BLA, who do you think will give it to you if you give up all your intellectual property? Because big pharma will ask you, where are your patents, where are your rights? But we were lucky enough that NIH actually funded all these phase 3 trial at the facilities, and they also told me we'll never do it again, it's too expensive. So that from now on, I may retain some of the intellectual property of future invention. But I think sharing with the world, like I was inspired by Banting and Best and Collett after inventing insulin that gave it up to the world for $1 nominal sale. And as I said, I don't pretend that this was an equally important invention, but that was really inspiring to me. And personally, I never charge a patient $1 in my entire professional life, and I hope to be able to continue to do so. The DRI, all transplants are free, and I hope we can continue to provide solutions that are not just safe and effective, but also affordable, because I would hate to see something that works without immunosuppression and then is charged hundreds of thousands of dollars and make it unsustainable for healthcare system or inaccessible for all patients who deserve a cure now. So we have to work on all the three legs of this important pillar that is like safety, efficacy, affordability, and we have to keep focusing on all three.
Michael, can I say something?
Please.
I just want to, I think most people probably do understand, but so I work on the science and I push a lot of things forward, but Dr. Ricordi is in one of those unique positions and has the passion and the energy to take this forward on all fronts. So sometimes the political part may not sound so comfortable, but without that kind of advocacy, without his absolute drive to make this happen, we wouldn't be sending, those letters wouldn't be getting sent now. This is something he's been working towards for a long time. And so it is important that I just wanted to recognize that, Camilo, and because it isn't always easy to talk about, but thank you on behalf of my family.
Now you make me emotional.
Really
I
important
cannot
part of
talk
it.
anymore.
Good, it's about time you got emotional. I always am.
Couldn't agree more. And thank you, Dr. Kenyon, for enhancing what Dr. Ricordi said. To either or both of you, what makes targeting the CD40 ligand pathway particularly promising compared with conventional immunosuppressive strategies used in islet transplantation?
Want me to take that one, Camilo?
Yeah, of course. I'm a surgeon. I'm not smart enough.
Yeah, I've heard that line. One of the reasons I was so excited about anti-CD40 ligand when I first heard about it is that the T cell, so that's the one lymphocyte I'll make you think about tonight, is the primary cause of rejection, also a major player in autoimmune disease. And for many years, the drugs that we have used don't even target lymphocytes specifically, but block cellular pathways that are common not only to the immune system, but other organ systems. So it may turn off the immune system, making you at risk for infection and cancer and having other side effects on other organ systems. I have a picture I often show of a surgeon friend of mine holding all the drugs that a transplant patient takes to counteract the fact that they're immunosuppressed, antiviral, antibacterial. So we were very excited about anti-CD40 ligand. Okay, so number one, on the T cell surface, there are other drugs newly approved that still, even if it targets the T cell, if it targets all your T cells, then you're still at risk for being over immunosuppressed. But this CD40 molecule, the way I've described it in the past is you meet someone, you think they're great, you have a conversation, that's really nice, that's the first interaction. So T cells, they see the transplant, that's the first interaction. But unless I call the person back and say, hey, I want to talk again, unless that T cell gets a second signal from that transplant, there's no immune response. And one of the earliest, there are many second signals, but one of the earliest ones is CD40 ligand. So CD40 ligand is the calling card for now the immune system to go ahead and attack the graft. Once the T cell upregulates CD40 ligand and sees the co-receptor on the transplant, you'll get a full-blown immune response. Now, instead of turning off your whole immune system, we're turning off the cells that are recognizing the transplant. And what we've seen over the years is that it doesn't make you, that viruses have a way around this, because someone could ask, well, what about other things like viruses? Will you fight those off? Yes, we've seen that it's been very effective. So that was number one, that it would be more specific. It limits inflammation, which is an issue when you first transplant islets, a very big issue because you can lose islets to inflammation when you put them in the liver. It also prevents B cells from making antibodies to the transplant. Antibodies to the transplant are a late cause of graft rejection. So it's preventing that T cell talk, it's preventing the B cell antibodies, it's limiting inflammation, and it's not diabetogenic, doesn't have adverse effects on other organ systems like the kidneys. So that
in a nutshell, as a surgical comment, in a nutshell, is amazing. It's a short answer.
Is the just a follow-up to that, is the CD40 ligand blockade currently being explored or used in other clinical contexts beyond islet transplantation? And if so, which areas of medicine are showing the most promising results?
Yes. Why don't you answer that, Camilla? Because there's back in the day, and you're more familiar with all those.
Well, no, it has been, we had a big discussion also with Eled on leadership because I'm not very diplomatic and I'm a little disruptive in my thinking. But when I found out about this drug from Anelixis, was a spin-off of the ALS Foundation that developed it to treat ALS, and I saw it after JP Morgan at a meeting to explore new potential breakthrough. And I said, wait a minute, you engineered the molecule to eliminate the side effect, maintaining the biologic function. We're back in business for diabetes and islet transplantation. And I said, we'll raise the money to make it happen for diabetes and islet transplantation. And then when the company became public, they said, well, kidney transplant is a bigger market, and they made the strategic decision to go in kidney transplantation. So now kidney transplantation has a 98% success rate at 1 year, and you don't have diabetes to begin with, so you don't see that big effect on metabolic and side effect. You don't see the same problem that you can have if you start having a little nephrotoxicity or organ toxicity. So I thought, and I'm so happy that now they are refocusing on islet transplant and diabetes. There are trials also in type 1 diabetes to intercept disease progression. Sanofi is having a similar molecule that they are testing in type 1 diabetes to stop progression of autoimmunity. There is potential use in all autoimmune diseases, and yes, of course, also in other organ transplant. But I think the one where you will have the best route to approval and show efficacy and safety in a very small number of patients will be islet transplantation. And then for sure, I'm confident will be expanded to many other indications in autoimmunity and transplantation as a whole.
We've had a couple.
Amazing.
I'm sorry, go ahead, Norm, please.
I said that's amazing.
We've had actually a few people add a question about kids. Their kids or other kids. When will we be seeing trials for under the age of 18? What does that look like moving forward?
Well, I think that anti-CD40 ligand is a good step in the right direction. I'm incredibly excited about Sonia Schrepfer new technology of gene editing to remove all immunosuppression, as well as the fast ligand microgel of high tolerance. If that can remove all immunosuppression after a transient immunomodulation of a few weeks or months, then is when you will start talking about expanding the indication to young adults and children. And unfortunately, I have to say, I'm so sorry we're all crushed, and I'm really sorry because we just lost the chief executive officer of High Tolerance that suddenly died for a heart attack or arrhythmia while perfectly healthy, and was so excited because we were just discussing how to move the technology towards clinical trials in the next year and what country, which geographic location, and he just left. So my condolences to the entire team and the family, but we cannot stop, and we find renewed energy even moving forward in his name. And I'm a strong believer in combination therapy. So I think that if Sonia Schrepfer and her team will move on gene editing to protect from autoimmunity besides allotransplantation, and the anti-CD40 ligand could provide initial help in this direction, and we may have high tolerance fast ligand microgel for a few days to increase the regulatory T cell at the transplant site. And also for the induction, now we have amazing new molecules like the human ATG anti-thymocyte globulin that is much with no side effects. You can redose, can target memory T cells that are important to eliminate in autoimmunity and promote regulatory T cells. We have so many things in the pipeline that are now reaching the clinical stage that I believe that the combination therapy will be allowing us to do it without immunosuppression and bring this to children, hopefully in the next few years. I don't want to put a timeline because it's since 1984 that I say in the next 3 to 5 years we may have something spectacular. And then we keep, I always say it could be less than 7 years or more than 15, but we have to keep working with the same focus, enthusiasm, and resilience, like the cure is behind the next corner. And I will never give up on this kind of strategy and philosophy.
But by the way, I just want to mention that several people, including a couple of physicians on this call, have expressed their support for your advocacy in terms of reclassification, and they want to know how they can help.
Well, I want to tell everyone that I'm incredibly thankful to Kathy Beth Hand and Devin Hall, because you have no idea. You have hundreds of patients complaining, say, I've been sick and with diabetes for 20 years, 30 years. When is the cure? When is the children? But very few people took steps like getting on the barricade and creating this very strong voice of advocacy to try to convince the Secretary of HHS to change this regulation. Just take one signature is scientifically unfounded the way that islets are regulated like right now. They are microorgan, they're comprising thousands of cells, they have a vasculature, they come from donors that are one different from the other. The first requirement of a drug therapy is that all the raw materials are the same for everything. And you have no idea how people and patients can move the needle, because if he's a transplant surgeon, if it's me or Piotr Inkowski saying, we want to change, and say, yeah, sure, you want to change because you want to work more, do more transplant at your transplant center. But we were at a meeting like last week where the people from the rare disease field said what make us a difference for us with FDA to allow new therapy to be tested are patient advocacy and the parents and the patient coming to FDA to the meeting to say this is what we live every day and why this is why we want a change. Like at FDA in the past, I was shocked when I heard like people that are no longer there, thankfully now, but is saying, why do you want to cure diabetes? There is no unmet need in diabetes. You have insulin, you have pumps, you have glucose meters. So what is all this fuss about the cell therapy? And then go check the statistic on the death on the waiting list for pancreas or islet transplant. They go from 5% to 30% death on the waiting list waiting for a transplant when you have these severe cases. So without scaring anyone, but each of us has examples in our families and centers and cities. And when I hear someone that says that there is no unmet need, we should be thankful. Of course, we're thankful there is insulin, that there are glucose sensors, that their day-by-day life has been improved. But if someone tells me that there is no unmet need, I think it's time to change a lot of things.
Thank you. And for those who did express their interest in supporting this cause, we will be reaching out to you after the call and connecting you with Dr. Ricordi to share how you can get involved more directly. A few more questions, if I may. In the context of islet transplantation, do you envision anti-CD40 ligand as a lifelong therapy, or is there a possibility that it could eventually support the induction of immune tolerance and reduce the need for chronic immunosuppression?
So we do have data that suggests. So I'm a little bit. I should tell you too. I'm a bit of a cynic, and I also think combination therapy is going to be very important. So we do have data that treating for a certain number of months, a certain period of time, that if you discontinue, now this is in a preclinical model, you do get long-term graft survival with fewer doses or none. We haven't actually had the opportunity to really test that out. We were working on that back in the 90s, doing experiments to specifically ask that question. So I do believe it is a tool in the tolerance induction toolbox. We need to do some more work. I think potentially with the fast ligand technology that Dr. Ricordi described to you, that could be one potential combination. But yes, I do think it has that potential. Camilla, you want to?
No, definitely. And especially in combination therapy, I see like that you could give, a lot of people make the mistake to say, I want to test this strategy without any immunosuppression. They start with no immunosuppression from scratch. And that is a recipe for failure, because at the beginning you have danger signals, inflammation, you have to get the islets revascularized. And the combination strategy, if you start doing gene editing like Sonia Schreiber is doing and others to decrease the level of making the islets stealth, and then you address inflammation at the beginning, giving some anti-inflammatory treatment, then you have anti-CD40 ligand that blocks co-stimulation, then you may have something that you can just need to treat with something mild, safe, and effective without side effect for the first weeks or month, and then progressively stop everything. And then after 3-6 months, you're off completely any drug and have islets surviving forever. And I think that is my hope. And one of the limitations is that companies don't like generally to work together or team up because they all think that they have the solution for everything. And then we see over and over again in trial net for blocking progression of diabetes. We spend decades doing single agent trial that all fail. And now, thanks also to Dr. Schuyler, and we convince everyone that now combination strategies are the way to go. And I think we show that in islet transplantation, and I think we'll show it for autoimmunity and many other indications. But I think we will be able to stop completely.
One question that came in is about the Vertex and Eledon trials. Can you, in as simply as possible, just describe the difference between the two trials and whether one learned from the other?
Vertex and the Eledon. So the Vertex is just standard immunosuppression, like the CIT NIH phase 3 trial with Prograf, tacrolimus, so it has the same side effect, the same diabetogenicity, because they wanted to start we wanted in the Vertex trial to show FDA requires show that you can do as good as islet transplantation with conventional immunosuppression, but Eledon is moving one step ahead because with Piotr and our collaborative effort, because you can show that now you can replace the most toxic anti-rejection drug with something that is more safe and equally or more effective. So then this is a ring a bell to Vertex saying, guys, you should either buy Eledon or collaborate with Eledon. Sanofi has similar molecules, and I think who will apply this kind of more advanced, safer and less toxic immunomodulation strategies to stem cell derived product will make the next big quantum leap also for stem cell derived product. And I think I'm very hopeful for so even the China group are looking at anti-CD40 ligand very carefully because they know so far they've been using Prograf as well, so they're like Vertex.
And Camilo, there was a
Please,
question
Dr. Kenyon.
about Myfortic. You know, we don't want to. The anti-CD40 ligand, as Dr. Ricordi has said, is replacing the Prograf, the FK506. There are still other agents that we clearly want to minimize the use of and get rid of, but it's a progression. This is a big step forward. So we're not trying to say that all of the agents that have some side effects have been eliminated, but certainly the most toxic one to the islets and other systems, and with some of these other advancements that we're studying and that Dr. Ricordi has specified, I think it will be possible to continue to remove those drugs as we move forward.
I know you've both had a very long day. Just we'll get through two more questions and then we'll call it an evening. And I assure you, any other questions that we haven't addressed, as I said earlier, we will reach out to you and provide answers. And Dr. Ricordi and Kenyon will both be involved in those answers. What is the greatest difficulty? Sorry, it just went away. What is the greatest difficulty for the success of encapsulated implants?
Well, I've been testing every encapsulated system and macro device since 1985, when Geraldine O'Shea and Tony San developed the first encapsulation system. And the biggest problem is that you introduce a semi-permeable barrier that blocks nutrients and oxygen diffusion, at least in part, and you have you have islets in a cage that, you have catabolic, you have metabolisms from the islets. You need to get not just oxygen and glucose to sense and respond with insulin. You need macronutrients. It's just enough to have a little reaction of one layer of fibroblast around the capsule and the islets inside are choked. So I'm not aware of any micro or macroencapsulation procedure that has been successful over a few months in large preclinical models or human clinical trials. Everything works in mice, of course. We have 440 ways to cure diabetes in a mouse, and the real, I wish everyone working with encapsulation, if we want a suggestion, move to something relevant preclinical and then do a pilot clinical trial if you want a real answer. But I don't want to say ever this will never work. I'm just saying I'm turning 69-year-old on April 1st, and I don't have time for green bananas, but I hope that some of the new generation will solve also the problem of micro or macro encapsulation, but not on my watch right now. I prefer gene editing and tolerance induction, personal preference.
Last question.
Was it too direct?
Never too direct, and don't go changing. That was well said. To both of you, and you touched on this, Dr. Ricordi, a bit earlier, but what do you say to families who've become a bit disenchanted with hearing for decades about getting closer to a cure?
I say you're right. I would just be incredibly upset on how slow and ineffective I've been doing, despite sacrificing my family that had to live the pain of not having me home while my children were growing up. But I'm not giving up if you're not giving up. And but if you prefer me to go fishing or open a restaurant, I'm ready to consider this option as well. But I think if you look back, it's always a long time when you're living day by day with a disease condition. But if you go back and see what we have achieved in these last decades, when we went from curing animal and mice to having now 20-year patient survival superior to insulin without immunosuppression. We have the new strategy replacing the most toxic immunosuppression. We have Piotr presenting 10 out of 10 patients in Barcelona off insulin without using Prograf. That was considered something that is absolutely needed for islet transplantation. So we are going definitely in the right direction, and I know it has been too long, and I take all the blame for what I'm concerned at the DRI on the 3 decades that I led the institute. But I'm never been so enthusiastic in my life as the progress that we did in the last 5 years. I said this before also when we finished the successful phase 3 trial, but is one step at a time, and the important thing is not to stop at third base. Thomas Starzl, that is the pioneer of transplantation and organ transplant, told me, I consider him my greatest mentor when I was in Pittsburgh and we did the first successful trial of islet transplantation in the world. He told me, remember, the biggest mistake that professor academic leaders do is stop at first base or second base, third base. You cannot stop until you reach the goal of what you impose yourself as your professional objective, to curing and helping one patient at the time, but not stopping until you find a cure for all.
Dr. Kenyon, any final thoughts?
I really couldn't say it better than that, except to agree that my family definitely did not see me very much. There have been moments of disenchantment. Of course, when you have diabetes from a very young age or even older, and you live with it day by day, and you watch your parents go off to work, I think that my father, who passed away 6 years ago, when my daughter was diagnosed, I was very upset because I kept asking the doctors for information. I wanted data. I wanted to understand what was happening. It was such a shock. And he said to me, well, most people don't want to know the data. I said, I want the data. And he turned to me and he said, there's 2 things my father said to me. One is, when it's you, it's 100%. Forget all the statistics. Now, I don't think there's anything truer than that. But he also said something that I have on one of my slides, and he meant it, because he never gave up until the end. Never give up, never give up, never give up. And I'm grateful to work with Dr. Ricordi and the people at the Institute to keep pushing forward. Don't give up.
Thank you. Thank you both. And I can tell you that everybody in the chat is shouting, never give up, and please don't go fishing. So we're all behind you.
I'm
Thank you
trying
both.
they were asking on the chat all the reason why we believe this can be reclassified by HHS, and is everything is there. Like, it is in the power of the Secretary of HHS of RFK Jr. now to write a letter and make an executive act saying islets are considered like organs, because it's been already established. It is already in the law that islets are sub-part of the pancreas, and the pancreas and sub-parts of the pancreas are considered organ and should be regulated like organ transplant. There is already UNOS is regulating pancreas and islet transplantation. Some people has raised the thing, if it goes out of FDA, it will be less safe, and instead is very much safe under the organ transplant, because this is a system that control the quality, the safety, the efficacy of the transplant programs. It's not that you allow everyone and their sister to do islet transplant, but you allow qualified academic center to perform them with a very strict regulation for patient safety, follow-up, transparency on all the data. There are registry that report, and you can go check every center by center what is the quality and the success rate that they can achieve, like you can do for organ transplant. And Israel is within his reach. He already did it for bone marrow, for cord blood, for composite tissue transplant. And it's just one letter and say islets are also now regulated as organ under OPT and UNOS rule, and make us competitive again and allow us to contribute to the search of a cure like other continents and countries are doing. I hope this. I'm very confident that this will happen this year. I don't see why not. There is no scientific rationale. There is no other reason why, without going into conspiration theories on what could be the interest that block the cure of islet transplantation. But I think it will happen. And thanks to people like Katie Beth and David Holt and many others. Also, I think we will make our voice heard. And I'm going to Washington May 26 already, possibly meeting someone.
Thank you. Thank you both for your time, your expertise, and your passion. And thank you, everybody on the call, for your participation, your terrific questions. A recording of the entire call will be available on our website, diabetesresearch.org, by the end of the week. And any questions that weren't answered we will be responding to you directly. So that's it for us. Thank you all.
I want to say Charlie Lawrence asking for the US restriction and all these points. I'll be happy to send an email. I was trying to put it in the chat, but I cannot find it right now.
And several others Dr. Ricordi asked as well. So we'll get you the names and we'll be able to share it with anybody who's interested.
We should maybe post it even on the website, answer to all the key questions that were asked.
Sounds good. Will do. Thank you, everybody. Have a great evening. Thank you for your participation, your time. Take care. Bye-bye.
Bye. Thank you all.